First, there was some last-minute switching around, and for those of you who are here to see Simon, I'm sorry you're going to have to wait 15 minutes through my talk. Hopefully it's not too painful. And second, I'm a really big fan of straightforward titles, but unfortunately in this case, it scoops the whole talk, and you've had a lot of time to look at it right now, so we'll get on with it. Okay, so in most animals, one sex typically remains in their natal group, while the other sex disperses to a new group. And in mammals, the sex that typically disperses is the males. Unsurprisingly, this dispersal event is quite stressful for the individual, the time during dispersal and entering a new group. I sort of like to think of it when you went off to university, right? It was really, really stressful to leave your home group and enter a new social group, right? Because there are many associated costs of dispersal, such as loss of past social relationships, high levels of aggression when you try and integrate into this new group, and hopefully this didn't happen to you at college, but higher levels of predation and higher rates of mortality due to that because of increased time spent alone. Okay, so we can all sort of agree that this dispersal event is very stressful for the dispersing individual. What we also know is that intense stressors like this can have both lasting and transient effects on the physiology of an organism. For example, in humans, you can have a lasting effect of an extremely traumatic stressful event, and this could manifest itself as post-traumatic stress disorder, which can have lasting downstream physiological consequences. Alternatively, there could be transient effects of a strong acute stress. For example, the loss of a loved one can stimulate some intense grief, which can also actually cause severe but reversible heart dysfunction. For example, this is also called broken heart syndrome, where EKGs and echocardiograms indicate that a heart attack is actually happening and it's imminent, but there are no obstructions that are found. Okay, so this is a transient physiological effect. And even in dispersal, there are actually both lasting and transient effects physiological effects on the organism who is dispersing. For example, in hyenas, post-dispersal males have elevated testosterone levels compared to pre-dispersal males. And this suggests that there might be some disinhibition of testosterone secretion that happens during the dispersal event. And as many of you may know, testosterone has potential immunosuppressive effects, so this could have some downstream immunological consequences. Additionally, there's also evidence that dispersal has transient effects on the physiology of an organism. For example, in Chakwa baboons in Botswana, both testosterone and the stress hormones glucocorticoids are transiently elevated around the time of dispersal, which can also bear some immunological costs. So my collaborators and I sought to sort of reconcile these different findings and these different hypotheses, and we wanted to understand how long and to what extent dispersal alters the physiology of an organism. And rather than focusing on just one or a few biomarkers as previous studies have done, we decided to sort of harness the power of whole genome sequencing and measuring whole genome gene expression. The advantages of this is that we're able not only to look at just multiple biomarkers in multiple systems, but we're also able to establish, which systems are affected by dispersal and for how long and so on and so forth. So we had two sort of hypotheses, the first one being that dispersal has a lasting effect on the animal's physiology, and here when I say physiology, I mean my outcome measure, which is whole genome gene expression, and so I'll use those two terms interchangeably. So in the first hypothesis, we can see sort of two phenotypes, predispersal males and post-dispersal males, where predispersal males will have different physiological profiles or gene expression levels compared to post-dispersal males. Here in the red, the post-dispersal males, and the predispersal males in blue. So this would be evidence of a lasting effect of dispersal. Alternatively, we can see that dispersal transiently alters the physiology of the organism. And if this were the case, we would expect to see that more established males, so individuals who've been in their group for much longer, either before dispersal or after they've integrated into a new group, will have different gene expression levels than peridispersal males, so males who are near dispersal. So here are the names of the types of groups that I'll be using, and that's going to be my jargon for the rest of the talk. Okay, so our study subjects were Rhesus macaques on the island of Cayo Santiago, which is about 1 kilometer off the coast of Puerto Rico. This colony is not a native colony. It was established, they were introduced there in the early 1900s, and they've been there ever since. And while they are by no means a native population, they do act fairly naturally for macaques on this island, and they exhibit some normal life history events, and they behave rather normally for macaques on the island. For example, male macaques on this island disperse between 3 and 6 years old, which is pretty standard for macaques. And importantly, not all males disperse from their natal group. So about 20% of the males remain in their natal group throughout adulthood. And this is important for the study because it makes it so that we're able to compare and contrast males who are both pre-dispersal and post-dispersal while age matching them. So we don't have the confound of age and dispersal. Okay? And so in this study, we studied 19 Rhesus male macaques on the island of Cayo Santiago. And for the 2 hypotheses, the groups were distributed as such in terms of lasting effects. The lasting effects hypothesis, we had 8 pre-dispersal males and 11 post-dispersal males. And for the transient effects of dispersal hypothesis, we had 6 peri-dispersal males, so males near dispersal, and 13 established males. Now the data that we collected were collected when our collaborators trapped the monkeys between January and March of 2012. When they trapped these monkeys, they quickly drew some blood into CPT vacutainer tubes, which isolate primary blood cells. So they isolate these white blood cells called peripheral blood mononuclear cells, which are composed of basically monocytes and lymphocytes. And we decided this population would be, this tissue would be the best tissue in which to test our hypothesis. One, because it's easy to obtain without destroying the animal. And 2, and more importantly, it's a primary immune tissue. And if we're looking at immunological costs or varying immunological costs of dispersal, we would expect it to manifest itself in these tissues. So after we isolated these blood cells, we then lysed them, harvested the RNA, and measured the gene expression individuals across all individuals using RNA sequencing. For the RNA sequencing, just some quick data, we generated over 600 million reads aligned to the Rhesus macaque transcriptome, and on average, across the 19 individuals, we had about 15.6 million reads mapped per individual, 2 exonic regions. But importantly, the take-home point from this slide is that we were able to confidently measure the expression of 10,161 genes. So we modeled the expression of these genes using a mixed effects model called GEMMA, which is good because you're able to control for many other compounds such as age and weight, and maybe when it was sampled, but more importantly, gene expression is heritable, so you're able to control for the relatedness structure, the population structure in your sample. And that's important for our study because some of these males came from the same group and they're likely to be related, so we wanted to control for that. Okay, so here's some results in terms of hypothesis 1. Well, we saw a few genes that showed the direction of having lasting effects of dispersal in terms of differentially expressed genes in pre versus post-dispersal males. However, when we looked at hypothesis 2 in terms of the transient effects, we saw many more genes that sort of popped up as being differentially expressed between peri-dispersal males and established males. And in fact, when we corrected for multiple hypothesis tests, none of the genes that showed differential expression between pre and post-dispersal males actually passed the false discovery threshold of 10%. But in terms of hypothesis 2, the transient effects of dispersal, we saw that about 1% of the genes, so 101 genes, were differentially expressed in peri-dispersal males versus established males, an FDR of 10%. So here's just a heat map showing those 101 genes. So our first question to ask with these data were, what are these genes? Well, since we're measuring a heterogeneous population of cells, these peripheral blood mononuclear cells, we could be seeing the signature of cell type specific genes and these cell types that are changing in proportion with respect to where that male is in terms of being peri-dispersal versus established. If that were the case, if the signature that we were seeing is mostly due to changes in cell type proportion, then it wouldn't be the case that dispersal alters gene expression profiles. It would just be that dispersal alters the relative proportions of these primary white blood cells. However, when I tested for enrichment of genomic signatures of cell type specific genes in those 101 genes, I found no enrichment for cell type specific genes, which suggests that the genomic signatures we see here are not due to changes in cell type proportions near dispersal. We also wanted to look at, are these genes also the same genes that are implicated in that are affected by chronically socially stressed individuals? And to do this, I drew on 3 data sets from humans, 2 from humans and 1 from macaques, where we're looking at low status individuals or chronically stressed caregivers. And I wanted to see if there was significant overlap between the genes that we found were differentially expressed near dispersal and the genes that were significantly affected by chronically stressed individuals. And in fact, we found that acute stress is not the same as chronic stress. These pathways are not the same. The genes are not significantly overlapping. And so then we decided to look at, rather than just a gene-by-gene analysis, we wanted to look at gene pathways. So we wanted to see what sets of genes, in other words, what biological pathways were affected by dispersal transiently. To do this, we conducted a gene ontology analysis, as I'm sure you've seen many people do at this conference. I used TopGo and their Higham method, which essentially takes into account the hierarchical structure of the gene ontology tree. And here's what we found. At a nominal p-value of 10 to the -3, we found 6 gene ontology categories that were upregulated, significantly upregulated near dispersal, and 6 that were simply downregulated near dispersal. And what I really want you to take away from this table is that you should notice that there is a strong enrichment for immune-related genes. So you can see that 8 out of the 12 processes that are significantly altered during dispersal or near dispersal are immune-related processes. So overall, we found that dispersal sort of transiently alters gene expression levels. We found no evidence for dispersal exerting a lasting effect on gene expression levels in male macaques. And evolutionarily, this makes sense because dispersal is a natural component of this organism's life history. So if there were detrimental long-term downstream consequences, it would be pretty maladaptive. And also, we found that the majority of the genes that were affected were immune-related genes. And this hints at the immunomodulatory effects of testosterone and the stress hormones glucocorticoids, which we know are elevated near dispersal. So overall, we found that while male macaques bear some immunological costs during the dispersal event, they return to a baseline and they're able to recover from these costs during more stable periods of their life history. And with that, I'd like to thank my collaborators and my funding sources.
Thank you. We have time for a quick question. Yep. Hey, no, that was great. I was wondering, since you have blood samples, if you have any data on whether the animals also had caught viruses or something, like as they're dispersing through the environment, are they getting infections which are leading to the increased immunological cost, and sort of whether, if this is known in general, whether animals are getting infected in things with dispersal, and that's kind of maybe one of the reasons why this happens.
Right, you would think that, I mean, and it could be sort of, you're thinking chicken and egg there. It could be that they're getting infected because they're immunosuppressed, or they're a little immunosuppressed because they're being infected. I don't know. The short answer to your question is, do we have those data? No. These data were collected in 2012 before I started my postdoc. I've been on the back end of this analysis. But that would certainly be something that we could look forward to doing follow-ups. I mean, one obvious question is, are testosterone and glucocorticoids elevated in these dispersing individuals? We don't really know in this sample, and that seems like an obvious next step.
All right.
Thanks.
Thank you once more.